All right, this is Mr. Price. We are going to work through the light-dependent reaction of photosynthesis on the thylakoid membrane. I'm going to draw on this as we go, so please note that the drawing is going to be amazing and you're jealous, I know. So first thing, this is the thylakoid membrane. I want to make sure that you realize that this terrible shape I'm drawing right now is a complete thylakoid. On the inside of the thylakoid is called the lumen. It's really hard to write with the mouse, so go with it. And on the outside of the thylakoid is called the stroma. Again, we're in a chloroplast in a plant cell, okay? So I want to keep this as simple as I can. So the light-dependent reaction is composed or breaks down into three separate parts. And if you can break these down into three separate parts, it's going to be easier to explain for everybody, okay? So I'm going to separate them based on this. Also, by the way, this picture's on your test and you have to be able to label it. So this is going to be one part of it's going to be Photosystem II, and it's going to be one part of your 3. The second part is going to be Photosystem I, which is this center part, and then the last part is going to be ATP Synthase. So what I've done is I've drawn lines here, and I'm going to talk about that part first. That's going to be my 1, okay? If I can get out of here. This is going to be my second part I'm going to talk about, and this is going to be the third part that I talk about, okay? So that should help you a little bit, all right? So again, anybody out here in the fans, if you need something, let me know. So I'm going to start with Photosystem II. They call it Photosystem II because we discovered it after we found Photosystem I. So 3 parts, starting part. Here's how I would go about saying this. The first thing that happens on the thylakoid membrane in the light-dependent reaction is that a photon of light, which is this right here, hits a chlorophyll A molecule. That's that green pigment right here, chlorophyll A molecule. And that molecule shakes or it resonates or it moves. And that movement creates enough energy to split the water molecule into 3 things. Those 3 things are hydrogen ions, which are now going to be these little red guys, hydrogen ions. It also releases electrons when it resonates and shakes the chlorophyll A molecule, that photon of light. And it also releases oxygen, and that oxygen leaves the thylakoid and goes up into the atmosphere, okay? So again, let me say it again. Photon of light hits chlorophyll A. I'm going to draw a terrible A there. That chlorophyll A resonates or shakes, and that shaking motion splits water. It splits it into electrons. Those electrons get into the membrane, thylakoid membrane, creating a negative, electronegative in the thylakoid membrane. The oxygen is released in the atmosphere, and the hydrogen ions are inside the thylakoid in the lumen, okay? I want you to bring your attention to this guy right here, this protein. This protein acts as a channel. And if you heard me a little bit ago, because this thylakoid membrane is negative and it has a whole bunch of electrons, remember when you were a little kid, you played with a bar magnet? Anytime you played with that magnet, you probably played that game where like, I'm not going to touch it and see if it'll draw the positive to it. The same thing's happening here. So I have a negative magnet right here in this protein, and that negative magnet, if there's any positives outside in the stroma, it sucks them into the lumen of the thylakoid, okay? So that's Photosystem II. Photon hits chlorophyll A, creates enough energy to split water into hydrogen ions, electrons, and oxygen is released. The negativity of these proteins in this electron chain pulls in any positives outside from the stroma. That is the light-dependent reaction Photosystem II first part. So I'm going to do something here. Nope, I'm going to keep doing this. Boom. All right. Okay, so my lines disappeared, so I'm going to put them back. We are right here now in Photosystem II. So in Photosystem II, there are, and I want to highlight that here real quick because I want to make sure. Photosystem II, which is between these 2 blue lines, is basically that electron is losing energy. It's falling slowly, and you kind of see that fall. It's losing its excitability. It's falling, if you remember back to the electron orbits, it's falling in those orbits. And so Photosystem I, all it does is it acts like a seesaw. that the photon of light hits the chlorophyll A again, right here, this A, and it re-excites that electron up to a higher orbit. So we have another photon of light, we have another chlorophyll A, it re-excites the electron, and simply all it does is it re-excites the electron so it can be packed away by this backpack. The backpack is called NADPH. That NADPH will now go out into the stroma to the Calvin cycle. Okay? So let me say that again. Photosystem I, photon of light hits another chlorophyll A, re-excites the electron, and NADPH picks that electron up and carries it away to the Calvin cycle. And those electrons are used to make sugars. Okay? So this is the part where finally the last part is this thing called ATP synthase, which I just put an arrow at. And I want the analogy in your mind to be an alternator in your car. And that alternator in your car spins when the car starts and it recharges batteries. So the ATP synthase spins, creating ADP into ATP, recharging ADP by adding a phosphate to make it ATP, okay? And the reason it does this is pretty cool. So we have a whole bunch of hydrogen ions. I'm gonna just put a whole bunch of H's inside of my thylakoid. And so because I have a high concentration of hydrogen ions, they wanna move from a high concentration to a low concentration in the stroma. And the other cool part about this is that in this drawing, and I'm adding that, remember that thylakoid membrane, super electronegative, it's got a whole bunch of, it's negative. And if I have a positive moving through a negative, it creates an electromagnetic field. And so it actually spins this protein. We have some electron microscope pictures of this protein spinning. And as it spins, it recharges ADP into ATP. So that's a light dependent reaction in the thylakoid membrane. Again, broken into 3 parts: Photosystem II, Photosystem I, and the ATP synthase. Thanks for watching. I hope that was helpful to all of you. Good day.